James and reference James music album 6 April 1928 Basic during this years interest in watching and matured into officers decided to learn genetics. This became possible when he received a fellowship for a graduate study. Became possible when he received a fellowship for graduate study geology at Indiana University in Newton, where he received his PhD degree in 1951, a study of the effect of hard X-rays on bacteriophage multiplication. X-rays on bacteriophage multiplication. He met Crick and discovered their common interest in solving the DNA structure. The first series of efforts was unsatisfactory. The second based upon more experimental evidence and better appreciation the nucleic acid literature. It resulted only in March 1933 in the proposal of the complementary double helical configurations. Francis Gary Compton Crick was born in July 1916 at Hampton, England. He studied physics at University College in London and obtained a BSc in 1937. He completed PhD in 1954 on this thesis entitled X-rays, diffraction, polypeptides and proteins. A critical influence in Crick's career was his friendship with J.D. Watson, then a young man of 23, in 1952 the proposal of the double helical structure for DNA and the replication scheme. Crick was made by FRS in 1956 in the honor of the Watson and to include the John Collins Barron's Prize of the Massachusetts General Hospital in 1959, the Lashkar Award in 1960, the Research Corporation Prize in 1962, the ECO Nobel Prize in 1962. Principal ingredients of variations. Have you ever wondered why an agent always gives birth only to baby elephant and not some other animal? Or why mango seed forms only a mango plant and not on any other plant, given that they do the offspring identical to their parents, or do they surely insist on some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other, or sometimes even so different? These are several related questions are dealt with scientifically in branch biology known as genetics. This subject deals with the inheritance as well as variations of characters from parents to offspring. Inheritance is the process by which characters are passed on from parent to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Humans knew from as early 8000 to 1000 BC that one of the causes of variations was hiding in sexual reproduction that exploited the variations that were naturally present in the wild population of the plants and animals who selectively breed and select for organisms that both describe desirable character, for example, through artificial selections and something and domestications from ancestors. Wild cows. We have well known Indian example Saigal cow. Wild cows. We have well known Indian breeds example Saigal cows. Indian Saigal cow Sahiwal cow is in Punjab. Sahiwal cow is in Punjab. Sahiwal cow in Punjab. We must have recognized that to our ancestors knew about the inheritance of the characters and variations. They had very little idea about the scientific basis of these phenomena. Punjaban must have recognized that our ancestors knew about the inheritance of the characters and variations. They had a very little idea about the scientific basis of these phenomena. theory of epigenesis. Character seed shape, seed color, flower color, pod shape, pod color, flower position, estimate. Seed shape, seed color, flower color, pod shape, pod color, flower position, estimate. Seed shape, seed color, flower color. देखिए seed shape, seed color, flower color, seed shape, seed color, flower color, pod shape, pod color, flower positions, stem height. Seed shape, round wrinkle, seed color, yellow green, seed flower color, violet white, flower color, pod shape, full constricted, and then full flower pod. You say full constricted, pod color green yellow, flower position, axial terminal, flower position, stem height, tall dwarf. Okay. 711, 454 एक क्रोमोसोम, मतलब 1457 4 क्रोमोसोम और 7 क्रोमोसोम, 4 कैरेक्टर, 7 कैरेक्टर आर प्रेजेंटेशन 4 क्रोमोसोम, 7 कैरेक्टर का 4 क्रोमोसोम. फादर जेनेटिक्स जे मेंडल 1822 में आए थे, 1884 में चल रहे थे. Mendel's law of inheritance, it was during the mid-19th century that Hedwig was made in understanding of inheritance. Gregor Mendel conducted hybridization experiments on garden peas over 7 years, 1956-1963, and proposed the law of inheritance in living organisms. During Mendel's investigations into inheritance patterns, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. He experiment had a large sampling size which gave credibility to the data he collected. Also, the confirmation of his inferences from experiment on successive generations of his test proved that his result pointed to general rules of inheritance rather than the unsubstantiated ideas. Mendel investigation characters in the garden pea plant that were manifesting as to opposing traits. Example, taller dwarf plants, yellower cage seeds. This allowed him to set up a basic framework of rules of governing inheritance, which was expanded on by later scientists to account for the diverse natural observations and the complexity inherent in them. Mendel conducted such artificial pollinations, cross-pollination experiments using several true breeding pea lines. A true breeding line is one of breeding line is one that have undergone. Mendel conducting such artificial pollinations, cross-pollination experiment using several true breeding pea lines. A true breeding line is one that having undergone continuous self-pollination shows the stable trait inheritance and expression for several generations. Mendel selected several 12 true breeding pea plant varieties and here which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated, full or constricted beginning, or yellow pods and tall or round figure. Stem height क्या भैया, tall round. Flower color का violet white. Flower position axial terminal, pod shape, full constricted, pod color का green yellow. Seed shape round wrinkle. Seed color yellow green. पेटल स्टिग्मा फिर अंतर को कट कर देंगे. मस्कुलेशन स्टेमन का रिमूवल ऑफ अंतर, मस्कुलेशन रिमूवल ऑफ एंथर्स. मस्कुलेशन मेल में है मेल और ये फीमेल से चटका देंगे अंतर, तो मस्कुलेशन है. फिर ट्रांसफ़र खोलेंगे पोलिनेशन, उसको चटकाओ, उसमें ट्रांसफ़र करो, फिर यहाँ स्टेम मेकिंग क्रॉस पी ओके petal stigma anther petal stigma anther stamen carpel petal stigma anther stamen carpel emasculation petal stigma anther stamen carpel petal stigma anther 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 petal stigma petal stigma anther stamen carpel remove करूँगा तो emasculation anther remove करूँगा then emasculations inheritance of one gene turn Johnson gene phenotype. Let us take an example of a such hybridization experiment carried out by Mendel where he crossed tall and dwarf pig plant to study the inheritance of one gene. He collected the seeds produced as a result of this cross and gave them to genetic plants. Thus, first hybrid generations. These generations are also called filial phenomenon. Roger Mendels observed that all the F1 body plants were tall like one of its parents known were dwarfed. He made similar observations for the other pairs of the traits. He found that the F1 always resembled either one of the parents and that the trait of the other parent was not seen in them. Mendel then self-pollinated and tall F1 plants and to his surprise found that in the filial generation some of the offspring were dwarf the characters that was not seen in F1 generation was now expressed the proportions of plants that were dwarf were 1/4 of the F2 plants were 3/4 of the F the plants were tall and blending that is all offspring were either tall or dwarf none were of in between height. Similar results were obtained with other traits that he studied. Only one of the parental traits was expressed in F1 generation while the F2 stage. Both traits were expressed in proportion 3:1. The contrasting traits did not show any blending at F1 of the stage. Allele term Batson. Allele the term from the Batson. The allele term Batson. Heterogeneous positions. Term hybrid should get the factor strategy. Okay, parental the tall of F1 generation tall surfing करेंगे भैया tall surfing F2 generation tall dwarf. Okay, based on observations Mendel proposed that something was being stably pushed down unchanged from parent to offspring through the gametes over successive generations. He called these things as factor. Now we call them as a gene. Genes therefore are the units of inheritance. They contain the information that is required to express particular trait in an organism. Genes which code for a pair of contrasting traits are known as allele. That is, they are slightly different forms of same gene. If we use alphabetical symbols for each gene, then the capital letters is used for the trait expressed at the F1 stage and the small alphabetical for the other trait. For example, in case of character of height, T is used for the tall traits and E for trait and T for dwarf and T are alleles of each other. Hence, in plants and their alleles for height would be tall or dwarf. Mendel also proposed that is a true breed. Tall or dwarf preferably the allelic pair of genes for height are identical or homozygous. Tall and tall respectively. Tall and tall are called the genotype of plant, while the descriptive them terms tall and dwarf are the phenotype. What then would be phenotype of plant and had a genotype tall. As mental form generated by one heteron child for tall to be exactly like the tall parent is appearance if certainly a pair of dissimilar factors, one dominates the other as in the F1 and the hence is called the dominant factor while the other factor is recessive. In this case, T for tallness is dominant over T for dwarfness, that is recessive. So, particularly the factors that he is studying. It is a convenient and logical to use the capital principle of a particular symbol of remember this concept of the dominance and this is to use T for tall and D for do not use T for tall and D for tall because you will find it as difficult to remember whether T and D are alleles of the same gene. Characters of no character or not alleles can be similar as in the case of the homozygote both tall and tall or can be similar as in the case of heterozygous TT. Since that TT plant is a heterozygous for genes controlling one trait and character height is a monohybrid and the cross between T tall and TT and TT is a monohybrid from the computation that is the same. Trait expressed without any blending in the F2 generations, we can either infer that when the tall and dwarf plant produce gametes by the process of the meiosis, all the alleles of the parental pair segregate from each other and only one allele is transmitted to the gamete. This equations of alleles is a random process and so there is a 50% chance of chemical either allele has been verified by the result of the process. In this way, the gametes of the tall TT plants have allele T and the gametes of the dwarf TT dwarf plants have allele T during fertilization to alleles T from one parent say through the pollen and T from the other parent then through the egg allele to produce jackets that have one T allele and one T allele. In other words, the hybrids that have TT since these hybrids contain alleles which express contrasting traits, the plants heterozygous the production of gametes by the parents and the formations of the jackets. The F1 and F2 plants can be understood from a diagram called a net square as one figure. It was developed by a British geneticist Reginald Silvernet. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written on two sides, usually the top row and left columns. All possible combinations are represented in boxes below in squares which generates the square output from the planet is cautious. The parental tall male and dwarf female. The planet also is the parental TT male and TT female. Plants gametes produced by them and F1 TT progeny plants. Therefore, plants generally equality in the symbols of female and male are used to denote the female eggs and female pollen. F1 generation is particularly the F1 plants with genotypes TT and self gametes with genotypes tall and T in equal proportion. Fertilization to express the pollen plants with genotypes T have 50% chance of pollen eggs of the genotype as well as of genotype T. Also, pollen grains of genotype T have a 50% chance of quality of genotype T as well of tall gametes, dwarf gametes, F1 generation, tall and self-limiting gametes, F1 generation split ratio. 3 ratio 1 genotypic ratio 3 ratio 1 phenotypic ratio 1 genotype 1 ratio 2 ratio 1 tall भैया phenotypic ratio phenotypic ratio phenotypic ratio phenotypic ratio 3 ratio 1 genotyping 1 ratio 2 ratio the assertion of random fertilization of 40 resultant diamonds with the genotype state or dwarf. From the kinetoscope it is easily seen that 1/4 of the random fertilization lead to the dwarf, 1/20 to the dwarf, and 1/4 of the dwarf of the F1 hypogenotype dwarf, but phenotypic characters in each tall at F2. 3/4 of the plants are tall, where some of them are tall, where others are not tall externally. It is not possible to distinguish between the plants with the genotypes tall and tall. Hence, within the genotype appears tall, only one characteristic tall is expressed. The characteristic or tall is said to dominate over the other allele or dwarf characteristics. Thus, due to this dominance of one character over the other, that all the F1 are tall through the genotype is tall, and in F2 or 3/4 of the plant are tall through genotypically 1 to 1 tt tall are only 1/4 tall. This leads to phenotypic ratio 3 ratio tall 1/4 tall plus 1/2 tall and 1/4 dwarf, that is 3 ratio 1, but if genotype ratio 1 ratio 2 ratio 1, the 1/4 1 ratio 2 ratio 1 1/4 1/2 1/4 ratio t dwarf is mathematically condensable. The agency of t tall dwarf in equal to frequency of the expression expanded at 2t plus 1/2 square equal to 1/2t plus 1/2t 1/2t plus 1/2t is equal to 1/4tt plus 1/2tt. Many such pollinated leaf-form plants are that dwarf plants continue to generate dwarf plants in 3/4 generations. Even to the genotype of the dwarf was much as dwarf. What do you think? And what do you think? Had got had self-pointed from the preceding paragraphs. It is clear that through the genotype ratios can be calculated using mathematical probability by simply looking at the phenotype of dominant traits. It is not possible to know the genotypic composition exactly. For example, where a tall plant from F1 F2 tall trans compositions cannot be predicted. Therefore, to determine the genotype of a tall and at F2 molecules in a tall plant from F2 with dwarf plant. This is called a test cross. In typically test cross and the organism B plants here showing a dominant phenotype and whose genotype is to be determined is crossed with the recessive parent instead of self cross. The progenies of a such cross can easily be analyzed to predict the genotype of the test organism. Shows the result of typical test cross where the violet color flower dominant over white color flower. Simple networks try to find the nature of some the test cross. What I should is using the gentle case cross can give general definition of a test cross. Consolidate. 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 Solid. मजबूत संकलन करना, संगठित करना, संगठित करना, कंसोलिडेट करना, कंडेंसेबल, कंडेंसेबल, धन्यभूत करने में, धन्यभूत गाढ़ा करने में, कंडेंसेबल, घनीभूत करने में, घनी धनी गाढ़ा करने में, घनीभूत करने में, गाढ़ा करने में, कंडेंसेबल, कंडेंसेबल, गाढ़ा करने, कंसोलिडेट, 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 क्रॉस करोगे तो बाई डोमिनेंट होगा, ये भी क्रॉस करोगे तो डोमिनेंट होगा, ये भी क्रॉस करोगे तो ट्रायंगल होगा भाई। तो रिसेसिव कहाँ से आ रहा है भाई? सेम प्रोसेस है भाई, डोमिनेंट टाइप जेन टाइप बनाना होगा। ओके, रिजल्ट और फ्लावर्स आर वायलेट डन, हाफ ऑफ फ्लावर्स आर वायलेट एंड हाफ ऑफ फ्लावर्स आर वाइट। ओके, represented a test process interpretations known for based on his observations on monohybrid cross. Mendel proposed to generals to consolidate his understanding of hybrid crosses. Today these results are the principle of law, principles or law of principle of law of trends, law of segregations, parallel law of Japanese industrial law of segregations, principle or law of principle of Mendel ने प्रपोज किया था प्रिंसिपल और लॉ principle or law of principles law of based on observations on molecular process. Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today these rules are called principles or laws of inheritance, 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 principles or laws of law of segregations, principles or laws of inheritance or law of segregation, principles or laws of and the second law of segregations. The first law of dominance and the second law of segregations, law of dominance. Mendel law of dominance. Mendel law of dominance. Mendel three exceptions: incomplete dominance, codominance, multiple allelism. 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 Incomplete dominance, multiple allelism. Incomplete dominance, codominance, multiple allelism. Law of dominance characters are common. First law दो लॉ किया था इसका लॉ ऑफ डोमिनेंस, लॉ ऑफ सेग्रेगेशन. लॉ ऑफ डोमिनेंस तीन एक्सेप्शन: इनकम्प्लीट कोडोमिनेंस, multiple codominance, multiple incomplete multiple incomplete codominance 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 multiple. लॉ ऑफ डोमिनेंस मेंडल ने दिया था जिसका नाम तीन एक्सेप्शंस हैं. इनकम्प्लीट डोमिनेंस, कोडोमिनेंस, मल्टीपल तीन एक्सेप्शंस हैं लॉ ऑफ डोमिनेंस में. इनकम्प्लीट डोमिनेंस, कोडोमिनेंस, मल्टीपल allelisms. Factors are controlled by discrete units called factors. Factors occur in a dissimilar pair of factors. One member of the pair dominates, the other recessive. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross. In the F1 and the expressions or both in the F2. It also explains the proportion of 3:1 obtained at the F2 allele alternate form of chain. What is allele alternate form of chain? Mendel law of purity of gametes, law of splitting of hybrid, law of segregation. This law is based on the fact that alleles do not show any gradient and that both the characters are discovered as such in the F2 generations. One of these is not seen at the F1 stage. Through the parents contain two alleles during gamete formations. The factors or alleles or pair segregates from each other such that a gamete receives only one of the two factors. Of course, homozygous parents produce all gametes that are similar, while heterozygous one produce two kind of gametes, each having one allele with equal proportion. देखिए मेंडल दो लॉ प्रपोज किया, एक लॉ ऑफ डोमिनेंस प्रिंसिपल और लॉ ऑफ इनहेरिटेंस बोल सकते हो. मेंडल ने प्रपोज करा है एक लॉ ऑफ डोमिनेंस जिसमें तीन एक्सेप्शन हैं: इनकम्प्लीट डोमिनेंस, कोडोमिनेंस, मल्टीपल है. तीन एक्सेप्शंस है लॉ ऑफ डोमिनेंस में. एंड देन सेकंड लॉ है लॉ ऑफ सेग्रीगेशंस, उसमें नो एक्सेप्शंस. ओके. इनकम्प्लीट डोमिनेंस when experiments on peas were repeated using other traits other plants. It was found that sometimes F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower, snapdragon or antirrhinum species is a good example of understanding incomplete dominance in cross between true building red flowered and the true breeding white flowered plants RR, and the F1 RR was the pink. When the F1 was self-polluted, the F2 resulted in the following ratios: 1 red flower, the RR pink, RR white, where here the genotype ratios were exactly as the would expect in any Mendelian monohybrid cross, but phenotype ratios changed from the 3:1 dominant recessive ratio. What happened was that R was not completely put over R, and this made it possible to distinguish RR as a pink from RR red, RR white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express particular trait. In the diploid organism, there are two copies of each gene. That is, as a pair of alleles. Now, these two alleles need not always be identical, as in a heterozygote. One of them may be different due to some changes that it has undergone, about which you will read further on, and in the next chapter modifies the information that particular allele contains. Let's take an example of a gene that contains the information for producing an enzyme. Now there are two copies of this gene. The two allele forms let us assume as in more common that a normal allele produces the normal enzyme that is needed for the transformation of a substrate. Theoretically, the modified allele could be responsible for production of the normal less efficient enzyme or dominant. The normal allele could be responsible for production of the normal less efficient enzyme or a non-functional enzyme or no enzyme at all. Parent generations में रेड होगा, RR रेड, RR वाइट, गैमेट्स करोगे भाई, F1 जनरेशंस RR पिंक हो जाएगा भैया, फीमेल RR हो गया, RR मेल गैमेट्स क्रॉस कराओगे 1:2:1, 1:2 जनरेटिक फिनोटाइप सेम होगा. ओके, पैरेंट जनरेशन रेट अगर रेड होगा, पेरेंट्स RR रेड, पैरेंट जनरेशन रेट R, पैरेंट जनरेशन रेट RR, parent generation rate RR, parent generations RR red होगा, RR white होगा, and R capital R small r white होगा. Then okay, theoretically the modifiers could be responsible for production of the normal less efficient enzyme, a non-functional enzyme or no enzyme at all. Dominant होगा, enzyme protein होगा, recessive. And in the first case, the modified allele is equivalent to the unmodified allele. That is, it will produce the same phenotype trait that is result in the transformations of substrate. Yes, such equivalent allele pairs are very common, but if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represents the original phenotype, is a dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. Codominance. Till now, we were discussing crosses where the F1 resembled either of the two parents. Dominance or was in between incomplete dominance. But in case of the codominance, the F1 generation resembles both parents. A good example in different types of red blood cells, and a good example is different types of red blood cells that determine ABU blood in human beings. ABU blood groups are controlled by the gene I gene. The plasma membrane of the red blood cells has sugar polymers that protrude. Protrude from its surface, and the kind of a sugar controlled by the IgM. Plasma red blood cells sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has three alleles: IA, IB, and I. The alleles IA and IB produce are slightly different from the sugar, with allele I does not produce any sugar. Produce slightly different form of sugar. IA and IB produce slightly different form of sugar. I does not produce any sugar. IA and IB produce slightly different form of sugar. IA and B produce slightly हल्का सा form of sugar करता है, लेकिन I does not produce any sugar. कुछ नहीं करता है, sugar कम भी नहीं करता है. Because human adequate organism, each person possesses any two of the three IgM alleles. IA and IB are completely dominant over the I, and in the other words, IA and I are present only IA expressed, because I does not produce any sugar. When IB and I present, IB expresses but when IA and IB are present together, they both express their own types of sugar. This is because of the codominance. Hence, red blood cells have both A and B types sugar. Since there are three different alleles, there are 6 different combinations of these alleles, of these three alleles that are possible, and therefore a total 6 different genotypes of the human AB blood types. How many phenotype possible? Table showing the genetic base of blood groups in human population. Allele form: parent 1 A, B, and I. Allele form to parent A, B. Genotype blood: normal codominance, normal codominance, normal codominance. Do you realize that the example of AB blood groups also provides a good example? Multiple alleles have, you can see that there are more than 2, that is there 3 alleles governing the same characteristics in an individual. Only 2 alleles can be present. Multiple alleles can be found only when populations studies are made. Occasionally, a single gene product may produce more than one effect. For example, star synthesis in pea seed is controlled by one gene. Star synthesis in P seed is controlled by one gene. Star seeds star synthesize in P seed. Star synthesize in P seeds is controlled by one gene. It has two alleles B and B. Starch synthesizes effectively in BB homozygous and therefore large starch and grains are produced. In contrast, BB homozygous jackets of less efficient starch synthesis and B smaller starch, and after maturation of the seeds BB are round and the baby seeds are wrinkled. Surround heterozygous. Present question on effect, for example, starts with P seed controlled by one gene. It has two alleles B and B. Starch is effectively BB homozygous. Their large starch are produced. In contrast, BB homozygous and produce smaller starch. After maturation of seeds, BB are round and the seeds are wrinkled. Heterozygous produce and round seeds, and so BB seem to dominate alleles, but the starch grains produced are of intermediate size BB seeds. So if starch grain is considered as the phenotype, then from this angle the allele shows incomplete dominance. Therefore, dominance is not autonomous feature of a gene or the product that it has information for. It depends as much of the gene product and the production of particular phenotype from this product as it does not the particular phenotype that we choose to examine. In case more than phenotype is influenced by the same gene, inheritance of two genes. Mendel also worked and crossed two plants that different into characters, as is seen in the cross between a pea plant that has seed with yellow color and a round shape, and one that had seeds of green color and wrinkle shape. Mendel found that the seeds resulting from the crossing of the parents had yellow color and round shape seeds. Here can you tell which of the characters is in the pair of yellow, green color and round wrinkle shape was dominant? Because yellow color was dominant over green and round shape, dominant over wrinkle. These results were identical to those that he got when he made separate monohybrid cross between yellow and green seeded plants and between round and wrinkled seeded plants. Let us use phenotypic symbols Y or dominant yellow seed color and Y for recessive green seed color, R for round shape seed and R for wrinkle shape seed shapes. The genotype of the parents can be written as RR and green. The cross between two plants written down as the figures 5.7. The genotype of the parents plants round yellow, round wrinkle yellow, the plants between the 5.7 the genotypes plants chemics are Neet unite a fertilization produces fruits when the Mendel self-having the effect plants found that a few plants yellow seeds and one effect had green. The yellow and green colors segregated in ratio 3:1 round and wrinkled seed shape, also segregated in 3:1 just like a monohybrid cross. F1 P generation round and yellow, RR brown, yellow, wrinkle or green. Gametes round, yellow, wrinkle, green. Different generation gametes होगा फीमेल का आर करवाओगे तो जाएगा राउंड येलो. Nutrition plus diabetes plus round yellow 9 round green 3 round green. Bovary chromosome. It is a bovary chromosome of inheritance. Bovary chromosome theory and bovary chromosome theory. Chromosome theory of inheritance, chromosome theory of inheritance, chromosome theory of law of independent assortment. In the dihybrid cross, the phenotypes round yellow, wrinkled yellow, round yellow, round yellow, wrinkled yellow, round yellow, wrinkled yellow, and wrinkled green, round yellow, wrinkled yellow, round green, and wrinkled green appeared in the ratios 9:3 ratio 9:3:3:1. Such ratios are observed of several pairs of the characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow, 1 green with 3 round and 1 wrinkled. This derivation can be written as follows: 3 round, 1 wrinkled, 3 yellow, 1 green, 9 round, 3 yellow, wrinkled green, 3 round green, 1 wrinkled green. Based upon the such observation, diagram was consecutive and different in 2 traits. Mendel proposed a second set of generalizations that we call Mendel's law of independent assortment. The law states that when 2 pairs of traits are combined in hybrid segregation of one gene, pair of characters is independent of the other pair of characters. The Punnett square can be effectively used to understand the independent segregation of 2 pairs of genes during meiosis and the production of eggs and pollen. They form RR round yellow plant round yellow. Consider the segregation of 4 genes R and R. 50% of the gametes have a gene R and other 50% R. Now beside each gamete has R or R, it should also have allele Y. Important to identify the segregation of 50% R. 4% of R is independent from the segregation of 50% Y and 50% of Y. Therefore, 50% of gametes has Y and the other 50% has Y. Similar 50% of R bearing gametes has Y and 50% of Y. Thus, there are 4 genotypes of gametes, 4 types of pollen and 4 types of eggs. The 4 types of R are Y, RY, and R, each have frequency of 50% or 1/4 of the total gametes produced. When you write down the 4 types of eggs and pollen on the 2 sides of the Punnett square, it is very easy to derive the composition jackets that give rise to the active plants. Although there are 16 squares, how many different types of genotypes and phenotypes are found? Note them down in the format given. Can you, using the Punnett square data work out the phenotypic issues at the active sites and fill the format given? is the genotype ratio. Also, non-ratio serial number genotype found in F2, the expected phenotypes. Certain chromosome chromosome certain Mendel published his work on inheritance of characters in 1865. Mendel published his work on inheritance of characters in 1865, but for several reasons it remained unrecognized in 1900. Mendel published his work on inheritance in 1865. Inheritance character 1865. Which is hundreds of letters, but several reasons remain unrecognized till 1900. Firstly, communication was not easy as it is now. In those days, Mendel's work could not be widely publicized significantly. Secondly, his concept of genes or factors in Mendel's words, the stable and schedules that control the expression of traits and of the pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation of apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematical to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, Mendel's work suggested that factors genes are discrete units. He could not provide any physical proof for the existence of factors that they were made of. Discrete. He could not provide any physical proof for existence of factors, so let's see that they were made of. In 1900, three scientists, Hugo de Vries, Cornelius Tischer, Correns, and von Tischerman, de Vries, Tischerman, Hugo de Vries, Carl Cornelius, Tischerman, Hugo de Vries, Carl Collins, independently discovered Mendel's result on his inheritance of the characters. Also by this time, due to advancement his microscopy that we are taking as three scientists were able to carefully observe cell divisions. This led to discovery of structure in the nucleus that appear to double and divide just before each cell division. These were called chromosome color bodies as they were visualized by Stephen by 1902. The chromosome movement during meiosis had been noted out. Walter Sutton and Theodore Roosevelt noted that the behavior of the chromosome was parallel to the behavior of genes and used chromosome movement to explain Mendel's law. Recall that you have studied the behavior of chromosomes during mitosis. Recall that you have studied the behavior of chromosomes during mitosis for gravitational divisions and during meiosis reductions divisions. The important things to remember are that chromosomes as well as genes occurs in pair two. The two pairs of a gene here are located on homologous sites of chromosomes. Given G2 bivalent meiosis 1 and meiosis 2 anaphase germs, meiosis and germ cells formations of chromosomes. Can you see chromosomes repeat and germ cells are formed? G1 में ऐसा है G क्रॉस बाइवेलेंट करके meiosis 1 चटकेगा भैया ट्रैक germs. Comparison between the behavior of chromosome and genes. Comparison between the behavior of chromosome and genes. Chromosome and chromosome between genes and chromosome between genes and chromosome between genes, chromosome between genes and chromosome between genes and chromosome functions of chromosome. Comparison between the behavior of chromosomes and genes. 1. Occurs in pair secrete at the time of gamete for between behavior chromosome and genes. Occur in pairs segregate at the time of gamete formation such that only one each pair is transmitted to gamete. Segregate at the gamete formation and only one of each pair is transmitted to a gamete. Independent pairs segregate independent of each other. One pair segregates independently to another pair. Can you clear? These columns A or B represent chromosome and which present gene and how did you decide? During the phase of meiosis, the two chromosome pairs are aligned at a metaphase plate independently of each other. Figure 5.9. To understand this, compare the chromosomes of 4 different coloring the left and the right columns. In the left columns possibility I orange and green is segregating together, but in the right hand column possibility 2 the orange chromosome is segregated with the red chromosomes. Possibility 1 possibility 1 long orange and short green. Long orange and short green chromosome and long yellow and short red chromosome at the same pole. Meiosis 1 brown yellow. Okay, possibility 1 long orange and short red chromosome, long yellow short green chromosome at the same pole. Meiosis 1 anaphase 1. Okay, meiosis 2 चटकेगा anaphase में ट्रैक जंप सेल्स में आई रेड येलो wrinkled green. Sutton and Beaufoy argued that a pairing segregation of pair of chromosomes the segregation of pair of the factors they carried. Sutton united his knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Following his synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas and Mann and his colleagues leads to covering the basis of the variations that sexual productions produce. Morgan worked with the tiny fruit flies. Morgan work करेगा tiny fruit flies. Explain other teach me. Tiny fruit flies. Drosophila melanogaster, which were found very suitable for the study. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle about 2 weeks. 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 They complete their life cycle. Drosophila melanogaster. Drosophila melanogaster. They complete their life cycle 2 weeks. They complete their life cycle 2 weeks. Drosophila melanogaster. Drosophila melanogaster. They complete their life cycle. They complete their life cycle. They complete their life cycle in 2. They complete their life cycle 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 in about 2 weeks. And a single mating could produce a large number of progeny pairs. Also, there is clear differentiation of the sexes. The male and female is easily distinguishable. Also, it has many types of hereditary variation that can be seen with low power microscopes. Drosophila melanogaster. Single mating could produce a large number of male and female are easily distinguishable. Also, it has many types of hereditary variation such as low power microscope. ड्रोसोफिला मेलानोगास्टर मेल छोटा होता है, फीमेल बड़ा होता है, अकॉर्डिंग टू एनसीईआरटी. Drosophila melanogaster. Complete the life cycle of about 2. Drosophila melanogaster. Complete the life cycle. Drosophila melanogaster. Complete the life cycle 2. Drosophila melanogaster. Drosophila melanogaster. Drosophila melanogaster. Complete their life cycle 2 weeks. About 2 weeks. Linkage recombination. Drosophila complete model with the experimental phylogenetics. Experimental phylogenetics model. Fruit fly Drosophila melanogaster linkage भी दिया था, term linkage ये दिया था. Linkage and recombinations. Morgan carried out several non-deliberate processes in Drosophila to study genes that were sex-linked. The process were similar to deliberate process carried out by Mendel bees. For example, Morgan hybridized yellow-bodied white-eyed females for brown-bodied eyed males and intercrossed their F1 progeny. He observed that the two-tailed genes did not segregate independently of each other, and F2 ratios deviated very significantly from the 9:3:3 ratio perspective. The two genes are independent. Morgan and his group knew that the genes were located on X chromosome section 5.4 ratios, and saw quickly that when the two genes in a deliberate process were situated on the same chromosome, the proportions of parental gene combinations were much higher than non-parental Morgan attributes this due to physical association or linkage of the two genes and coined the term linkage to describe his physical association of genes or chromosomes, and the term recombination to describe the generation of non-parental combination. 5.11. Morgan and his group also found that even when genes are grouped on the same chromosome, some genes were repeated. Linked shows in other shows higher recombinations. For example, he found that genes white and yellow are very tightly linked and shows only 1.3% recombination. White and miniature white shows 7.2% of recombination system Alfred. Miniatures also. Ring shows 37.1% system Alfred S31 frequency recognition between genes paired in the same organism, distance between genes, and mapped their position of the chromosome to the genetic maps. Are extensively used as starting point in the sequencing of the whole genomes, as was done in the case of human genome sequencing, project described. Yellow white recessive, female recessive, yellow white female yellow white recessive, 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 female yellow. Female yellow white recessive, 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 white miniature. White into yellow. Painted 98%, 28.7. Recombination 1.3%. 62.8%. Recombination 37.5%. Linkage relative lamp was connected by more than cross-issues crossing. Logic cross-issues crossing head over the contact is representing the person subscript. Note the strength of level between Y and W is higher than the modern orogenic inheritance. Hair color, height, skin color, eye color. Orogenic Mendel's studies mainly describe those groups that have distinct alternate forms such as flowers color, which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across gradient. For example, in humans, we don't just have a tall or short people as two distinct alternatives, but whole range of possible heights. Such traits are generally controlled by three or more genes and thus called as polygenic traits. Besides the environment of the multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example for this. In polygenic trait, the phenotypic reflect the contribution of each allele, that is, the effect of each allele is additive. To understand this better, let us assume that three genes A, B, C control skin color in human, with the dominant forms A, B, and C responsible for dark skin color and the recessive forms A and B and C for light skin color. The genotype in all the dominant alleles A and B and C will have a darkest skin color, and that with all recessive alleles A and B and C will have the lightest skin color as expected. The genotype with three dominant alleles and three recessive alleles will have an intermediate skin color in this manner. The number of each types of allele in the genotype would determine the darkest lightness of the skin in an individual. Neurotropy. We have so far seen the effect of gene on a single phenotype trait. There are, however, instances where a single gene can exhibit multiple phenotypic expressions. Such a gene is called neurotropy. The underlying mechanism of neurotropy is in most cases in the effect of a gene or metabolic pathways which contribute towards different phenotypes. An example of this is the disease phenylketonuria, which occurs in humans. The disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydrolase single gene mutation. This manifests itself through phenotype expressions characterized by mental retardation and reductions in hair and skin pigmentation. This manifests itself through phenotypic expression characterized by mental retardation and reduction in hair, reduction in skin pigmentation, reduction in skin pigmentation, reduction in skin pigmentation, sex determination. Sex determination. The mechanism of sex determination has always been pursued before the genetics. The identical clue about the genetic chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observation made in a number of insects lead to the development of the concept of the chromosome basis of the sex determination. In 1891 could trace a specific nuclear structure all through spermatogenesis in few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Hanking gave the name to this structure as the X-body, but he could not explain its significance. Further investigation by other scientists led to the conclusion that X-body hanking was in fact chromosome, and that is all. And that is why it was given the name X chromosome. It was also observed that in a large number of insects, the mechanism of sex determination is of the X type, that is all X bear an additional X chromosome beside the other chromosomes. Autosomes. On the other hand, some of the sperms bear the X chromosomes, whereas some do not. X fertilized by sperm having an X chromosome females and those fertilized by sperms that do not have an X chromosome become males. Do you think the number of chromosomes in the male and female are equal due to the involvement of X chromosome in the determination of sex? It was designed to be Z chromosome and the rest of the chromosomes were named as the autosomes. Grasshoppers is an example of the extra type of sex determination in which the males have only one X chromosome besides autosomes, whereas females has a pair of X chromosomes. Females has pair of X chromosomes. Grasshopper is a example extra type of sex in which the males have only X chromosome beside the autosomes, whereas the females have X. These observations relate to the linguistics and number of species, understand the mechanism of the sex determination in a number of other insects and mammals, including an XY type of sex determination is seen where both the male and female have the same number of chromosomes among the females and X chromosomes, but its counterparts is distinctly smaller and called the Y chromosomes of females. Determination that both male and females have same number of chromosomes among male X chromosomes present, but counterpart is still smaller and called Y chromosome. Females have X chromosome. Both males and females bear the same number of autosomes. Hence, the males have autosomes plus XY, female have autosomes plus X. In equinox and Drosophila, the males have X and Y chromosome, whereas a pair of X chromosomes beside autosomes. In above the descriptions, you have studied about two types of sex determining mechanism, that is XY type and XY type, but in both cases, male produce two different types of gametes, either with or without X chromosome, or some gametes with X chromosomes and some with Y chromosome. Such types of sex determination mechanism is designed to be example of heterogamity in some other organisms. Example, birds and different mechanisms of sex determination is observed. In this case, the total number of chromosomes is same in the males and females, but different types of gametes in the term of the sex. Chromosomes are produced by female, that is female heterogamity, in order to have distinctions with the mechanism of the sex determinations described earlier. In the two different sex chromosomes of female birds has been designed needed to be ZW chromosome. Female birds ZW chromosome. In these organisms, females have ZW chromosome, where males have a pair of Z chromosome beside the autosomes. It's fat. sex determination in it has already been mentioned that the sex determination mechanism in case of humans is Y type of output. Out of 23 pairs of chromosomes present, 22 pairs are exactly the same in both males and females. There are in the autosomes a pair of X chromosomes are present in the female, whereas the presence of X and Y chromosomes are determinant of the male characteristics during the spermatogenesis. Multitude types of gametes are produced. 40% of the Y chromosome besides the autosomes also produce only one type of X chromosome. Locally probability of fertilization of among the sperm carrying either X or Y chromosome. In case of fertilization fertilizes with the sperm carrying X chromosome, zygote develops into female XX, and fertilization with the Y chromosome carrying sperm resulting into the male sophia. It is evident that it is genetic makeup of the sperm determines the sex of the child. It is also evident that each progeny is almost 50% could be either male or female child. It is unfortunate that in our society women are blamed for giving birth to female children and have been ostracized, ill-treated because of the false notion. Blamed female को blame किया जाता है या ostracized. Ostracized. With notion, false notion. धारणा विचार false notion, then ostracized. Ostracized by script किया जाता है ostracized. Sex determination is a honeybee. The sex determination in honeybee is based on the number of seeds of chromosomes and individual receives an offspring formed from the union of the offspring from the union of the sperm and eggs developed for the female queen or worker, and unfertilized eggs developed as the male draws by means of parthenogenesis. This means that the males have half number of chromosomes than that of female. The females are diploid having 32 chromosomes and males are haploid, that is having 16 chromosomes. This is called haploid sex determination. The system has a special characteristic features as well. Males spermatosis. They do not have a father and cannot have sons, but have grandfather had a grandsons. How the sex term mechanisms different in the words in the call. पैरेंट 32 मेल फीमेल्स में 16 मेल में एप्लॉयड हो रही है मियोसिस करेगा माइटोसिस करेगा मियोसिस करेगा मियोसिस 16 मेल 16 हो रही है फीमेल का सही साफ नोट करेंगे तो मियोसिस करेगा मतलब टूट जाएगा माइटोसिस करेगा ना फीमेल और मेल टूटेगा मियोसिस करेगा माइटोसिस भैया ये म्यूटेशन ना चाहिए sudden change in the genotype cytosine oncogenic virus that cause cancer physical X and Y chromosome various gamma rays chemical biological mutation at gene level point mutations chromosome structure chromosome mutation is a phenomenon which result mutation is a phenomenon mutation is a phenomenon which result in alternation. Mutation is a phenomenon which result in alternation of DNA sequences and consequently result in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variations in DNA. As we will learn in chapter 6, one DNA helix runs continuously from one end to other end, each chromatid in highly supercoiled form. Therefore, loss, deletions, or gain insertions, duplication of segment of DNA result in alternation in chromosomes. Since genes are known to be located on chromosomes, alternation in chromosomes result in the abnormalities or aberrations. Chromosomal aberrations are commonly observed in the cancer cells. Aberrations of cancer cells. In addition to having mutations also arise to the chain in single base pair of DNA. This is known as point mutation. A classical example of such sickle cell anemia, deletion and insertions of base pair on DNA causes friendship mutations. See chapter 6. Mechanism of mutations beyond the scope of this discussion at this level. However, there are many chemical and physical that induce mutations. These mutagens of radiations can cause mutation in organisms. It is a mutation. Gender disorder degree. पहले तो सिंबल पढ़ लो तुम तो पहले सिंबल पढ़ लो. Physical X-ray, chromosome, UV rays, gamma rays, chemical biological mutation at gene level, point mutations, chromosome structure. Mutation at gene level, point mutations, chromosome structure, mutation aberrations. Chromosome number, chain number, mutation symbols are male square, male female sex and unspecified male square में male का सिंबल स्क्वायर में male का स्क्वायर में male का स्क्वायर से male का स्क्वायर से 6 unspecified section is section 6 is 14 is 6 section is for section specified section 6 is 44 section is section is specified section section is specified section affected individuals mating between relatives continuous mating cousin and cousin parent age of the children below in order of birth left to right above and children below in order parent with male child affected disease parent with male child disease 5 unaffected offers and 5 unaffected offers 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 5 unaffected 5 unaffected offers 5 unaffected 5 unaffected offers 5 unaffected offers 5 unaffected 5 unaffected 5 unaffected 5 unaffected 5 unaffected offers 5 Sorry. Last question. The principle of maintenance, the botanical maintenance of such maintenance resources can be addressed in family life. Delays is the most common right prevail in system. Cystical cystic turnover is important to motion treating questions may also link to the subconscious case of revealing that questions of quantum recessive traits discussed teacher transmission from unaffected carrier female to swarm of male has been widely studied in this disease. A single protein that is part of the class A of proteins including blood is affected due to this unaffected dosing but will result in non-stop bleeding. The head corresponding carrier for hemophilia may transmit the disease to focus on the possibility of becoming hemophilia is extremely rare because mother of such a female has to be at least carrier and the father should be hemophilic and will be unviable in the literacy of the family. Putting with clinical shows the number of hemophilic days and events as she was a carrier for the years considering disease and autonomous transmitted can be transmitted from parents to other efforts meaning that the parents are carrier for gene or electrovirus the disease of control by a single paraphernalia is controlled by a single paraphernalia out of 3 possible gene types and the images individuals show the disease from the types of 2 gene types which means we will separate blindly unaffected but they are carrier of the disease as they are 50% probability of transmissions of the gene due to the progenitor gags. Six position of the beta globin chain of the hemoglobin molecule, the substitution of the globin protein resulting to the single level substitution at the 6th codon the beta globin chain from undergoes polymerizations and low emissions causing a change in the shape of the abusive by continuous like structure. Phenylketonuria, the inborn metabolism is inherited as that is on receptor and the affected so that's an enzyme that converts the amino acid phenylalanine into tyrosine as a result of this phenylalanine is accumulated and converted into phenylpyrrolacid, and other derivatives. Accumulation of these in brain result in mental retardation of these during the course of its poor absorption in the kidney. The same mechanism is also present in the necessary blood disease causing from parents to parents are unaffected carry gene of heterogeneous therefore the defect could be due to accumulation of mutations which ultimately result in reduced rate of synthesis of one of the globin chain and final beta chains that make up hemoglobin. This causes the formation of hemoglobin molecules resulting into hemoglobin which is characteristic of the disease. Can also make them classified according to which chain the hemoglobin is affected in the production of alpha chain is effectively production of hemoglobin chain is affected. Alpha thalassemia controlled by two closely linked genes HB1 and HB2. Yes, molecules produced maximum gain and 11 movements across mutations of which is 1084 from sickle cell NMH to quantitative problems of synthesis to hemoglobin molecules. Later is qualitative problems of the synthesis and correctly falls improvement. Sickle cell qualitative thalassemia quantitative thalassemia qualitative is sickle cell chromosomal disorder and are caused due to the absence of excessive rearrangement of the normal chromosomes, failure of segregation of chromatin during cellular cell cycle. One cycle resulting in the gain of loss of chromosomes called aneuploidy, for example, Down syndrome resulting in the gain of extra 21. Similarly, Turner syndrome resulting due to loss of X chromosome. One famous failure of cytoplasmosis after telephone stages resulting in this normal set of chromosomes. This fragmentation is polyploid exclusion is often seen in the plants. The total number of chromosomes in normal humans in age 46 reports. Out of these 22 chromosomes have 6 chromosomes. Sometimes obviously either can be additional individual may lack one of any one pair of chromosomes. These situations are known as trisomy or monosomal chromosomes respectively. Such as situation leads to very serious consequences. There is Down syndrome, Turner syndrome, scleroderma syndrome, all examples of chromosomal disorders. Down syndrome, DPKT. Chromosomal DPKT, Down syndrome, Turner syndrome. Disorders level down the diabetes and the flat back of teeth, many lips, fingers, tips, palm, creasing, increases of temporal flat and clinical tongue, congenital heart disease, carotid. Down syndromes, Downs, the cause of genetic disorder, presence of additional copy of the one chromosome in the trisomy. This disorder first described by Landon Down in 1966. The affected individuals of stage and the small forearm and partially open 5.16. Palm is broad and characterized palm increased physical feels, psychomotor and mental development is retarded. Filter syndrome This disease is also caused due to the presence of an additional copy of X chromosome resulting into characteristics. Such an individual has a overall masculine development, however, the feminine development of breast that is gynecomastia is also expressed. Such an individual characteristics are tall stature, full, nice characteristic of stature, only characteristic disorder due to sex chromosome confusion against Turner syndrome, such as disorder is caused due to absence of one, such as disorder is caused due to the absence of one. Turner syndrome, such a disorder gynecomastia trisomy. Connected to the investigator also. Some genetic is a branch of biology which deals with the principle of inheritance and its practice mechanism. The parents' morphological and physiological features attracted attention of the many biologists. Mandel was the first to study systematically studying the pattern inheritance of composite characters when composed Newton's which are today referred to as a mental law. He proposed that the factors later known as chain stability impairs first yellow follow different pattern line different first generation form, second form, and so on. Some characters are dominant over others. Components characteristics when factors are in heterogeneous conditions. The recessive characters are all expressed in homogeneous conditions. The characters never blend in heterogeneous conditions. The recessive characters that was not expressed in heterogeneous condition may be expressed when it becomes homogeneous. Hence, characters are between formation. Elements of segregation not all. Character shows two dominance. Some character shows incomplete and so codoms in mental study. Principle to characters to be it was found that the factors independently certain combining permutations and combination of independence different combinations of games are theoretically represented in a square tabular form when it is called the factors now known as gene equations regulating those characters are called the genetic and physical expression of the characters are called mental. After knowing that genes are located on the chromosomes and good correlations was drawn between Mendel's laws segregation and thus assortment of chromosomes during analysis. The Mendel's law were extended in the form of chromosome theory 20 years later. It was found that Mendel's law of independent assortment does not hold true for the genes that were located on the same chromosome. These genes were called as antigens. It was related located genes assorted together and distantly located genes due to recombinations. Event-led linkage maps therefore corresponded to the result of genes of chromosome. Many genes were linked to sexes also called as sex-linked genes, and two genes are male and female were found to have certain ways which are where which are also did chromosomes which were dependent to sexes were named as sexes chromosomes influence and male female has 22 years of protogens appear 6 chromosomes of male has 22 pair of protogens and pair of 6 chromosomes of chicken 6 chromosomes it is different as children genetic material mutations change the single base pair range because they mean to change one base in the gene beta gene. Mutations can be studied by generating a period of families of mutations in one gene and the etc. Chromosome polyploidy or gene a subject of chromosome number inequality. This help in understanding the mutational basis of disorder. Down syndromes is due to the presence of chromosome 21 here, whereas there is an extra chromosome 21 here. Consequently, the total chromosome becomes 46 chromosomes and chromosome missing and 6 chromosomes is not there. While X chromosome missing and chromosomes X not and clarified symptoms condition XXI. These are easily understood as common types.